My name is Kulanova R.М. I am biology teacher and I am working in the secondary school gymnasium number 17 named after Ivan Trubetsin. I want to introduce your attention my lesson. The lesson topics features of the structure and functions of non-membrane, single membrane and double membrane organoids in the cell, the main components of the cell. Learning objectives. Explain the structure and function of cell organoids as seen under an electron microscope. Firstly, I want to speak about cell. Cell biology is the study of cell structure and function, and it revolves around the concept that the cell is the fundamental unit of life. Focusing on the cell permits a detailed understanding of the tissues and organisms that cells compose. A microscope is a device designed for obtaining magnified images, as well as measuring objects or structural details that are invisible or poorly visible to the naked eye. The combination of technologies and methods of practical use of microscope is called microscopy. Firstly, speak about cells. Cells are the smallest form of life, the functional of structural units of all living things. Your body contains several billion cells organized into over 200 major types with hundreds of cell-specific functions. Some functions performed by cells are so vital to the existence of life that all cells perform them. For example, cellular respiration. Others are highly specialized, for example, photosynthesis. And now you want to introduce to do this task. You should write all the organelles of the cell. What do you know? After this, you should do next task, complete the task on the site learning apps org and perform the task which is called the organelles of the cell. You can see the addresses of the link. And next slide about organoids. Organoids or organelles are permanent cellular structures that ensure the performance of specific functions by the cell. Each organoid has a specific structure and performs certain functions. There are one membrane organoids, A. Single membrane, B. Double membrane, and C. Remembrane organoids or organelles. And now I want to introduce next slide, and this slide to consist the task. Working in pairs complete the glossary task, and you can see the glossary. And next slide I speak about membrane organelles. We divide on single membrane and double membrane organelles. Single membrane organelles consist lysosome, endoplasmatic reticulum, Golgi apparatus, and double membrane organelles. These are chloroplast, nucleus, and mitochondrion. Next slide, I speak about endoplasmatic reticulum. Endoplasmatic reticulum is present as an incarnation of tubules that are connected to the nuclear membrane in eukaryotic cells. There are two types of ER based on the presence or absence of ribosomes of them. Rous ER with ribosomes attached to the cytosolic phase of endoplasmatic reticulum, and thus this is involved in protein synthesis. Smooth ER, which lacks ribosomes and has a function during lipid synthesis. Golgi apparatus or Golgi complex or Golgi body. The Golgi apparatus is a cell organelle mostly present in eukaryotic cells, which is responsible for the packaging of macromolecules into vesicles so that they can be sent out to the site of action. Lysosome. Lysosomes are membrane-bound organelles that occur in the cytoplasm of animal cells. These organelles contain an array of hydrolytic enzymes required for the degradation of various macromolecules. There are two types of lysosomes. Primary lysosomes contain hydrolytic enzymes like lipases, amylases, proteases, and nucleases. Secondary lysosome formed by the fusion of primary lysosome containing engulfing molecules of organelles. And about to speak vacuole. Vacuole are membrane-bound structure varying in size in cell of different organisms. Structure. The vacuole is surrounded by a membrane called tonoplast, which includes fluid containing inorganic material like water and organic material like nutrients and even enzymes. These are formed by the fusion of various vesicles. So vacuoles are very similar to vesicles in structure. Mitochondria. Mitochondria are double membrane bound cell organelles responsible for the supply the storage of energy for the cell. The oxidation of various substrates in the cell to release energy in the form of ATP, adenosine triphosphate, is the primary purpose of mitochondria. Structure. A mitochondrion contains two membranes with the outer layer being smooth, while the inner layer is made with folding and finger-like structure called cristae. Chloroplast. Next organelle. Chloroplast is a type of plastids that is involved in photosynthesis in plants and algae. Chloroplast contains an essential pigment called chlorophyll necessary to trap sunlight for the production of glucose. Structure. It is a double membrane structure with its own DNA, which is inherited from the previous chloroplast. And next organelles is nucleus. The nucleus is a double membrane bound structure responsible for the controlling all cellular activities as well as a center for genetic material and its transferring. It is one of the large cell organelles occupying 10% of total space in the cell. It is often termed the brain of the cell, as it provides commands for the proper function of other cell organelles. A nucleus is clearly defined in the case of a eukaryotic cell. However, it is absent in prokaryotic organisms with the genetic material distributed in the cell. And now I want to speak about remembrance organelles. The first, ribosomes. Ribosomes are ribonucleoprotein containing equal parts RNA and proteins along with an array of other essential components required for protein synthesis. In prokaryotes, they exist freely, while in eukaryotic, they are found either free or attached to the endoplasmatic reticulum. Next, cytoplasm. The cytoplasm refers to everything present inside the cell except the nucleus. Structure. The cytoplasm consists of a cytosol, a gel-like substance that contains all the matter, cell organelles, smaller cell-like bodies bound by separate membranes, and cytoplasmatic inclusions insoluble molecules that store energy and are not surrounded by any layer. The cytoplasm is colorless and has about 80% water along with various nutrients required for the cell. And now I offer for my students work on rows and pairs, where the first pair prepares a question about non-membrane organoids. The second pair prepares a question about single membrane organoids, and the third pair about double membrane organoids. And I offer the assignment 3, complete assessment 3, complete the task on the Google Classroom site, and you should open the lesson 14. And on this lesson, on this slide, you can see homework and reflection, slide about reflection. You should write what I learned, what remains unclear, what you need to work on. And I want to speak. Thank for your attention.